Right. Good afternoon, everyone, or hello. I suppose it might be morning, depending on where you're joining us from. Thank you all for coming today and for joining this virtual open day session where I'm going to describe our BSc Infection and Immunity. I'm Richard Milne. That is my photo on the screen. I'm a professor of virology here at UCL, and I am also the program director of our BSc. I'm joined today by Gemma Freeman. Gemma is waving hello. Gemma is one of our current final year BSc students, and she is going to be available later on in this session to answer your questions and maybe talk a little bit about what she's been doing this year for her research project. So, oh, that's interesting. So I'm actually in this building, this red brick building that you can see right here. This is the Cruciform building. I'm right just about there. And this is the main campus of UCL in central London. And it's very well located. It's a great place to be within walking distance of lots of excellent museums, galleries, and entertainment opportunities. So we're very well placed. On the agenda today, I'm going to start by talking about some science. This is meant to be an open day talk about our program, but our program is anchored on the science that we do in this division, the science of infection and immunology. And so I think it's appropriate to start by giving you a sense of some of the science we do. So I'll tell you a little story about some work that was done in this building, actually, at the start of the COVID pandemic 5 years ago. And then I'll make some broader conclusions from that and ask some perhaps slightly provocative questions. And then I'll run through the structure of our BSc program quite briefly. I don't want to sort of spend ages listing lectures and modules, but I'll try and give you a sense of what you would cover and what your experience would be. I'll give you a sense of what the sort of outcomes of the program are and where our students have ended up. And then we will open up for questions. If you have questions, please use the Q&A function. There should be a Q&A option on your webinar control panel. Feel free to type in questions at any point if you think of anything. So you can ask questions about the science, about the program, or about infection or immunity in general. And we'll try and get through all of those in the latter part of the session today. Right, so some science. I'm a virologist. I'm a professor of virology. And so I'm interested in viruses. And this is a very basic schematic of what a virus is. It's some nucleic acid, DNA or RNA, genetic material, the genome, wrapped up in a protein container. Obviously, that is a bit simplistic. That's a cell, a very simplistic depiction of a cell. You guys will be very familiar with cells from your biology studies, so you know all these parts. You could probably tell me what a mitochondrion does or how a nucleus works, and so on and so forth. As you probably know, viruses are unique in biology. They're a sort of law unto themselves because they're not cells. They're really the only biological entity that is not cell-based. And on their own, essentially, they're inert. They don't do anything. They can't move. They don't have sort of homeostatic mechanisms. And so whilst they're not cells, they need cells. And viruses need cells in order to make more viruses. And when a virus goes into a cell, essentially what it does is it subverts all of this kit, all this machinery that you're very familiar with, all of the cell biology, and turns the cell into a factory for creating more viruses. And that's deeply fascinating. And aside from any sort of disease or epidemic interest that viruses hold, because they're all adapted and evolved to exploit cells to make more viruses, they're fantastically good experimental tools. If you want to find out how something works in a cell, stick a virus on and look what the virus does. And that's a really, really good tool for discovery. And we do that a lot. And that's a very important part of our program. So when a virus goes into a cell, it has to do all of these things. It has to get in, has to express its genes, replicate its genome, build new particles, and then it has to transmit to the next host. And essentially, the more viruses you can produce, the better your chance of transmitting. And this is what we call a replication cycle. And this is HIV, but it could be any virus, really. And you'll be aware that essentially it's a bad thing to have a virus. You're better off without it than being infected. And so through evolution, cells have become very, very resistant to infection. They have mechanisms and systems in play to sense that they are infected and to intervene and block the infection at almost every step, actually, almost to paralyze the capacity of the virus to repurpose the cell. And one of the central mechanisms that cells use to signal that there is a virus about and to trigger other cells to switch on all of their antiviral mechanisms is a protein called interferon. And you may have heard of interferon. It's quite a well-known protein, and it's really, really important in determining the outcome of an infection. So here's that cell, but it's not on its own now. It's part of a group of cells. And let's say this is your nasopharynx. Let's say this is COVID. So the first cell gets infected. The infection is sensed. Interferon is produced. And crucially, that interferon is released from that cell, and it can travel to adjacent cells, and it can switch them into antiviral mode as well. So they are actually in their antiviral state before they've encountered the virus. And so the poor virus, when it's produced by this originally infected cell, it lands on this cell and it's dead in the water because it encounters all of these inhibitory mechanisms. And this is how we control infections. When you get infected with a virus and you feel a bit sort of fluey, this is what's happening. Interferon is driving that response and giving you that feeling of being quite sick. So next time you're feeling sorry for yourself with flu or the common cold, you can at least reflect on the fact that actually that's a signal that you're doing the right thing and you're getting rid of the virus. Now, we still have viruses. They still exist. So how is that possible? If we have all these fantastic antiviral mechanisms, how do we still have viruses? And the answer, of course, comes back again to evolution. Just as cells have evolved to be antiviral, so viruses evolve to be able to evade and suppress all of those antiviral mechanisms that cells present. And so you have almost a tit for tat. Cell evolves, virus responds. Cell evolves, virus responds. And that goes on almost forever. But that turns out to be really, really important in considering how viruses actually play out when they enter a population. And a really good time to study that is at the start of a pandemic, when you have a new virus and a completely naive population of hosts. And so that's what happened 5 years ago, when SARS-CoV-2 popped up. This is Lucy and this is Lorena, and they are working in one of our high containment labs, just upstairs actually in this building. And this was the very first experiment they did. So the virus arrived, we got the virus in about March, I think, or February of 2020. And the first thing you do when you get a new virus is you grow it. So this is a plate of cells. Each of these little sort of purple circles is a nucleus. And these are human airway epithelial cells. And this virus has been engineered so that it expresses a protein that glows green under fluorescence microscopy. And you can see that actually even just 5 hours after infection, there's one green cell, but within 10 hours, there are quite a few. And within 24 hours, the whole cell sheet is infected. So that's pretty exciting, actually, to do that with a new virus at a time when it was driving this emergent pandemic. So you may remember, although it was a long time ago, that the pandemic started in about sort of January, February 2020, went through a wave, then there was a bit of a pause in summer, and then there was a resurgence towards the end of that year. And there were headlines like this that popped up. There was a mutant virus, a new strain of COVID. Originally, it was called the Kent variant because it popped up in Kent. But in fact, we now call it the Alpha variant. And what the guys upstairs set out to do was work out what it was about that new virus variant that allowed it to transmit more effectively and effectively take over from the original virus that popped up in December or January. And the first thing you do when you get a new virus is grow it. And here's the key experiment. That virus arrived in this building on Christmas Eve 2020. So poor old Lucy and Lorena were setting this experiment up and spent Christmas Day and Boxing Day upstairs in the labs harvesting these 24 and 48-hour time points. But this is what we call a growth curve. So this is time in hours, and this is a log scale of the amount of virus, the number of copies of genome actually. Here's 1 million, 10 million, 100 million. So big numbers. And the blue one here is the original virus that popped up in January. And the red one is the new alpha. And you can see that it's replicating faster. It's about 24 hours faster, actually, than the original virus, and it goes a bit higher. So that's consistent with it taking over the world, it just transmitting better. But we want to know why or how that's happening. And to do that, we have to remember the interferon system and the antiviral mechanisms that are in play in the host. So remember this picture. Remember that the new virus is going to encounter the same defensive systems that the original one did. So you consider now, ask yourself, what might be going on here? What would a virus do that would allow it to replicate faster and better and to a higher level than its predecessor in the face of this quite effective response? So what Lucy and Lorena and co did was quite a simple experiment, actually. They reasoned that the really important component of the host response was that protein interferon. And so they took, at a single time point, just here, they sampled and worked out how much interferon was being produced in the infected cells as a measure of the response. And with the original virus, the blue one, here are the results. This is the amount of induction of interferon. You see quite high induction. 2 separate experiments to make sure that it's all correct. But with the alpha virus, you can see that it's way, way less. And so what that shows is that this virus has evolved to be better at evading and suppressing this really pivotal host response. And it's that adaptation to evade that allows the enhanced replication, allows the enhanced transmission, and essentially allowed this virus completely to replace the original virus globally in a very short span of time. So that was published in this paper. This is a scientific paper. This is in Nature. This is a really high impact paper. And there is the figure I've just described to you. So it's just one component of a bigger story. But what's super interesting is that it turns out that this was not a one-off. And if you look at what SARS-CoV-2 has done over the years, it's come in waves, and that's shown here. This was the original virus. Here's the first wave, Alpha. That was then succeeded by Delta. And then we have Omicron, and now we've got different waves of Omicron, all indicated by these colors. And here's the time schedule. And what's remarkable is that almost all of these new variants of CoV-2 have independently evolved to evade and suppress that antiviral, what we call innate response, just like the alpha virus did. So that's obviously a really, really important determinant of whether a virus can replicate and persist in a population. How well can it actually overcome the responses that are mounted? And we think that this is important, not just as a sort of historic way of documenting what's happened with COVID. We think that we can actually use this as a way of risk assessing future pandemic threats. So can we use a combination of the latest type of AI thinking and AI-based database analysis and the lab science that I've just told you about to work out which of the viruses in the jungle or in the caves in bats are the most dangerous? Which ones have the best capacity to evade human responses? Because we think those are the ones that are going to present the most pandemic risk. Can we actually synthesize viral genes and do those experiments before those viruses take off? Not growing the virus, just the individual genes. And so can we prepare vaccines or drugs for viruses that haven't jumped into humans yet? And that's a very real possibility. And that's a sort of future trajectory of the research that I've just described. And it's the sort of thing that lots of virologists are thinking about very, very seriously at the moment. Right. So that was the science part. I hope that you found that interesting. If you did find that interesting, then that is a good marker, I think, that this degree program would be a good choice for you. Frankly, if you didn't find it interesting, then probably this is not a good choice of degree. And you might be better off thinking about a broader bioscience program. But let's now spend a few minutes just outlining the structure of our BSc, the sorts of content that we cover, and then we'll stop and we can have a discussion and you can ask questions. So the aim really is to present and to deliver the science of infection and immunity as far as we can through current research. Now, that's not, you can't do everything that way. There are some basic things that you just have to learn. But in the specific infection and immunity content of the program, we do try and use current research, our current research, to inform and direct the teaching. And we want to do that because that's where the excitement is, right? The excitement is in what we're poised to discover, the questions that we're asking at the moment. And so we want to sort of encourage a sense of curiosity. You've got to be curious to do science. Science is about finding stuff out, and curiosity is the sort of fuel fundamentally that drives humans to ask questions. We want to encourage creativity. Science is a very creative enterprise. You have to come up with new approaches, new questions, new interpretations, new explanations. That's all very creative. And I suppose the product of science is new knowledge. And the process that gives rise to that new knowledge is discovery. So in one word, the job of a scientist is discovery. And we want to give you, as students on this program, the opportunity to really have a go at that, to access the state of the art and to try and discover stuff. And I think, most of our students get the chance to do that. And through doing that, you learn to think like a scientific investigator. So thinking in a way that is based on asking questions, formulating experimental approaches to answer those questions, creating data, evaluating and interpreting that, placing it in the context with other evidence to form new knowledge. So it's a 3-year BSc program. And like most BSc programs, it starts broad and gets progressively narrower and more focused. And the first year is very broad, and it's essentially teaching you how the human body works. And it takes a system approach to that. So rather than taking a sort of more traditional biochemistry, physiology, anatomy, genetics type approach, which would be what you would encounter in, let's say, a biosciences degree, it takes a systems approach: cardiovascular system, respiratory system, muscles, and so on and so forth. But in the end, it's still about learning cell and molecular biology and how these fundamentals come together to operate. The modules that you all do are listed here. And this is taught mostly at the Royal Free campus of UCL. This is the Royal Free Hospital, which is a few stops up the tube line from where I'm sitting. And it's taught jointly with a series of other programs within the Faculty of Medical Sciences. In year 2, there is some ongoing sort of systems type approach where they're addressing molecular medicine and some other aspects of the broad human disease. But increasingly, there's a focus on infection and immunity. And again, you can see the modules here. We have one specific infection module, one specific immunology module, and one module here that is just for I&I students. That's the journal club, where each week, as a cohort, you go through a current or a recent scientific paper and really start to get to grips with current thinking and the state of the art. And we have these sort of vertical themes which we want you to be thinking about from year 2 onwards. One is cutting edge. Science is not a discipline of history. Science is about what we're finding out right now. And it's very important that you're aware of that and that your whole approach is informed by that. Communication is super important. You could make the greatest discovery since Einstein, but if you can't effectively communicate that to the world, both in the written word and in the spoken word, then it's going to be of limited value. So we put a lot of time into helping you learn how to communicate science effectively. And critical analysis really underpins this whole business. This is not just accepting stuff. Just because I tell you a thing doesn't necessarily mean you should assume that it's right. You should say, well, why? Why do you say that? What's your evidence for making that assertion? And this is what I mean by when I said earlier, training you to think like a scientific investigator. I tell our students that over the years of this program, they will transition from being receivers and memorizers of information to being processors of information. And that's the pathway that we seek to funnel you through over the 3 years of this degree. So the final year of the degree is the culmination of that, where we focus completely on infection and immunity, on the research that we're doing and on the research that you're doing. In that year, the cohort of BSc students is joined by a cohort of our medical students. All UCL medical students do in their 3rd year what's called an integrated BSc, and they have a number of options to choose from, and one of them is immunology and infection, and that is the year 3 of our BSc program. So we have a joint cohort. We treat that as one group. It's all mixed up, and really you couldn't come up with a better group of people to be studying a scientific discipline at the interface of clinical medicine. So everyone does the same in the first term. We do 3 modules, immunology, an infection, and a pathology module. And everyone does a research project. We talk to students individually about their interests and match them with a host research group that is in line with their interests. And they do a research project. And hopefully Gemma will give you a 1-minute summary of what she's been doing. And then you also choose 2 modules from this list. So you can be quite broad. You can do an immunology and an infection module, or you can decide you want to do all immunology or all infection. Doesn't matter. Various combinations are available. The point about this year is that we're teaching about what we do. There's no universal fixed curriculum. We only have modules that cover stuff that we're researching. So, for example, there's no parasitology module on here because we don't have anyone in our division who's doing work on that. And we think that for a research-based program, it would be inappropriate. So we teach about what we do, and we genuinely focus on our research and your research through the project that you do. So what's the outcome? What are you going to get if you do this program? There are some obvious outcomes. You will definitely have a good understanding of the role of infectious agents and the immune system in disease, but also in health. Remember, your immune system is doing its stuff when you're well as when you're sick. Going back to the earlier years, you'll definitely understand at some level how the human body works, how the organs and systems work, but also you'll have an understanding at the cellular and the molecular level of the mechanisms that are in play. You will have real insight into current issues, current thinking in infection and immunology and beyond. And that will be particularly focused in the area that your project covers, and also in the area or areas that are captured by the optional modules that you choose. But universally, you will have familiarity and fluency with the language of discovery research and the concepts that are in play in that enterprise at the highest level. And in terms of skills, critical analysis, data interpretation, confidence in oral and written communication, and all of the sort of ancillary skill sets that go with that. How to manage data, how to design experiments, how to frame a question, how to make an argument. Those are all really essential skills. And really they are together what you might describe as the scientific method, which is something you will become familiar with. So what about the next steps? I mean, this seems a long way in the future for you, but just to give you a flavor of it, students from our program have gone, some of them go straight to do research as PhD students, either at UCL or elsewhere. A common path is to go and do further study as a postgraduate student, perhaps a master's degree or a Master of Research. Some students prefer to go and actually get a job doing research as a research assistant. That can be a path to further study, or it can be a path to a PhD. Some of our students don't want to do necessarily hands-on lab-based research, but want to stay in sort of allied careers. So there are many, many routes open to you in the sort of broader field of medicine and science, pharma, consulting, and some go sort of very off-piste. We've had one student go and do law. And I think, I mean, I say here on this slide, almost anything you want. And perhaps that's slightly flippant, but actually I do genuinely believe that. And my sort of evidence in support of that is the idea that the skills I just outlined and described as the scientific method are actually fantastically transferable. And you think of that student going to interview at a law firm, and they would say, well, hang on, what are you doing here? You've got an infection immunity degree. This is a law firm. And she would say, well, yeah, but let me tell you why I'm a really good candidate. I've got really good communication skills. I'm really good at dealing with complex, sometimes contradictory data sets, resolving those, providing a clear, concise argument. I'm good at dealing with complex ideas. All of this scientific method, if you present it right, can open doors beyond the lab. So I think far from being a sort of niche specialist degree, I suppose content-wise, it is somewhat that, although I argue that infection and immunity are simply a window onto the breadth of all bioscience and biomedicine. But actually, I think it's the antithesis of a niche degree, because of the skills that it's delivering to you and because of their universal deployability. So if you want more information about the university or the program, your first stop should be the prospectus, which is linked here. You can get information about the entry requirements and so on. The standard offer is on the screen there. I would encourage you to visit UCL. The campus is open. You can turn up anytime and have a wander around, or you can book a tour, which would probably be given by a current student, and there's a web page for doing that. There are also 2 open days coming up at the end of June, when you can come and listen to talks from program directors like me, but also other talks, and you can visit exhibitions and so on. I don't think you can sign up for them yet, but you can register your interest. But I'd really encourage you to do that. It's very important to get your feet on the ground if you can, and get a sense of what this place is really like. Okay, that is all I'm going to say about the program. So it's now over to you to ask any questions that you have, either about the science or about the general degree program. And I'll reintroduce Gemma, who is one of our current final year cohort, and she will be able to answer your questions as well, and I will defer to her in most matters. But whilst you're considering whether you have any questions, perhaps I can invite Gemma to maybe give a 1-minute summary of what her project was about, and also maybe just give us a sense of how you ended up on this program and how you found it.
Okay, so I guess I can start with how I ended up here and then jump into my project.
Yeah,
Um, yeah, so basically, I guess I was quite interested in immunology during like my biology and school days. And I was like, okay, I could do a program in biology and see what happens. And then I found this program and everything looked quite interesting. And it turned out really well. I'm
good.
like really happy that I like chose this. Um, yeah, I guess one of the things that was like um that like happens is you tell people that you're doing a degree in infection and immunity, and everyone's like, oh, that's so niche. And you're like, well, yeah, I guess, but I've learned about so much like different stuff, you know? Yeah. And then as for my project, it's like quite focused on my interests. So if this doesn't sound interesting to you at all, that's like totally okay because it's what I'm interested in. But I'm quite interested in bioinformatics. Um, I'm working in a lab that focuses on T cells, which are like one of the cells of your adaptive immune response. And so the T cell has this special receptor called the T cell receptor, and that's how it recognizes foreign things in your system. And basically, I could take your blood and I could sequence it, and I could have a look at your T cell receptors, and I could see all sorts of things. Like I could see what kind of diseases you faced, and stuff like that. One of the really cool things is that if I took a sample from you today and I took one from a month from now, and then 2 months and 3 months, and I compared them, and all of a sudden in one of the samples, there's like this massive change in your T cells, and all of a sudden they're completely dominated by one T cell receptor. That could give me an inkling that, hey, maybe you have cancer or you have some sort of autoimmune disease, which I think is really cool that we could be able to tell that just from sequencing your blood.
Yeah, me
Um,
too.
yeah, but one of the issues with that is that just from taking multiple samples, I'm introducing variability. So between two samples, nothing could have changed. You could have been like completely healthy. But just because I'm taking two separate samples, I'm sampling and I'm sequencing them, they could look really different. So my project was trying to kind of characterize what that difference is when nothing else is changing. Yeah.
Fantastic. So actually, that's a great example. Gemma has gone in a sort of, I guess, slightly mathematical, computational direction. Um, and her project very nicely illustrates the way that we can now do that. So we're by no means limited to doing lab-based science. And the scope, I mean, you'll all have heard of the AI revolution that's going on. I sort of hinted about it in my beginning of my talk when I said, can we use that for risk assessing pandemics? The opportunity and potential for combining the sorts of data sets that Gemma is working on with the extraordinary sort of analytical potential that AI approaches offer is mind-blowing, frankly. It's mind-blowing in terms of the discovery opportunity, but it's also mind-blowing in terms of the potential for therapeutic innovation. You know, once you understand, as Gemma is contributing to understanding, what actually is regulating a T cell response, then instantly you have a new handle on manipulating that and deploying it therapeutically. So I hope that's made the point that there is a lot of scope beyond going into the lab and doing pipetting. So I'll answer some of these questions. We've got 4 questions so far. Leah says, do students ever specialize in a type of disease or vaccine that they have an interest in? Well, Gemma has just evidenced that yes is the answer. So let me give you the big picture, and then maybe Gemma can comment. Yes is the answer to a degree. If your project, let's say you were really interested in I don't know, autoimmune disease, diabetes, we would try and fix you to do a project on diabetes. That would be a very dominant part of your experience, and you would have the option of also choosing our autoimmune disease module. So you could make this degree significantly into a diabetes degree if you wanted to. Some students do that. Some choose not to do that to keep things broad. But there is certainly a great degree of opportunity for you to specialize. If you're interested in virology, choose virus options, do a virology project. If you're interested in T cells, choose a T cell project and do the immunology optional modules. So yes, there is. But Not everyone does that. You don't have to do that. You can do this. As Gemma said, she chose this degree because she was just interested in this stuff. She wasn't specifically interested in disease X. So some people start broad and stay broad, and some people just get the bug and want to go and do a particular thing. And we try to encourage both of those things. Nadia, does the course also cover using viruses for benefit, such as oncolytic viruses? We don't have a major focus on that because there isn't anyone in the division doing exactly that, although there's one lab at UCL that's doing that. But we do have a cancer module, and we also have a therapeutics module, which touches on those things, but not in huge detail. Will, can I apply for both BSc and medicine? Yes, you can. That is possible. There are no constraints on what you can apply for. Tina, thank you for an insightful presentation. How do you compare this with other top programs in terms of length and breadth? Well, I would say, I assume you mean programs in infection and immunology. I would say that the broad content is going to be pretty similar. There's a certain sort of core knowledge, if you like, that you will get wherever you do this. I think what sets or what distinguishes us, this program, is the people that are here delivering the content, because the content is influenced and colored by what we think and what we are doing. And so that, in a sense, is what differentiates it from other places. I would say also that the assertion I made about really anchoring this program in research is real. I mean, is that a fair comment, Gemma? Do you think? What's been your experience of the way we link the research to the teaching?
Yeah, I've thought so. I mean, I always come into lectures and it's scientists telling you like, this is what I'm doing. Like they give you a bit of background and they're like, this is what I've done, this is what I'm doing. And it's really nice because everyone's really passionate because obviously it's like what they want to spend their lives studying. Everyone's really passionate and talking about what they're interested in and new stuff.
Mm,
And also I find that sometimes I'll be like doing research for a coursework and I'll be like looking at a paper and I'm like, hey, that guy gave me a lecture.
right. So what I should be clear about this. What I don't mean when I say research-based, I don't mean that it's just a load of scientists rolling up and giving their research seminars. That's not it. I mean, that there is an element of that. What I think I really mean is that what you get is a perspective, and a sort of level of insight that has been gained by many years of active engagement in research in that field. And so that sort of mindset is what gives this degree its particular flavor. I'm afraid, Tina, the second part of your question, what's the cutoff for IB? I do not have that in my head, but I know that it is on the prospectus entry page right next to the A-level requirements. So you can access that very easily and straight away. Okay, Leah, again, how often do you have to change the curriculum to include new or research, especially in year 2? Yeah, it's a good question. University is a bit different from school in that, in school, certainly in the UK, there is the curriculum and you all have to study that because you all have to do the A-level exam. And in order to pass the A-level exam, you have to know all the stuff on the curriculum. We don't have a curriculum as such because there isn't a national exam. We do the exams ourselves. And so the content of our course is really defined by what we think you need to know in order to meet the objectives that I've outlined. In other words, in order for you to be able to get to grips with the state of the art in current research. So the answer, how often do we change content? All the time. I can tell you with absolute truth and certainty that, let's say you joined this program in, October 25, you would be starting your year 3 in October 27. I have no idea what we would be teaching you in the virology module then, and I can tell you for sure that a lot of it won't yet have been published or discovered. So the answer is we continually update the content to reflect the latest thinking, the latest findings in the field. And that's what you would expect us to do, I think. Do we get a certificate to provide evidence of attending this webinar? I don't think so. I don't know. I don't think so. If you wanted to become an immunologist, is taking this degree a better route than taking a standard medicine degree? Okay, so it's very important that you understand that this is not a medicine degree, right? Medicine as an UCAS topic is what you study if you want to be a doctor and look after patients. This is a science degree. So if you're actually asking, is this a better route than doing a broader, more conventional biomedical sciences, biosciences degree or whatever? I think it very much depends on what you actually want to do. I think if you come to UCL and do biomedical sciences, for example, you have the option of choosing lots of the modules that we run in infection immunity. You might have the option of doing your project. You can switch to our degree for the 3rd year. So there are many routes into a career path as, let's say, an immunologist. I think if you are absolutely certain at this point in your career that that's what you want to do, then choosing this degree is quite a good choice because it gives you in-depth exposure early. On the other hand, there is an argument for staying broad and sort of sampling some other topics because it is quite early in your career as a scientist, and it may be that you actually want to do something slightly different. So I think a degree like this is obviously a good route into immunology, but it's not the only route into immunology. So I think the way to address it is to think about, do you want to really focus early, or would you prefer to start broad and have a bigger range of options? If you do this degree, you're definitely going to be doing infection immunity. That we don't have options to do genetics or whatever. So I think that's the decision. It's about how focused you want to be. But certainly, if you're set on a research career in immunology, then this is a good degree to take. So I think I've answered that question. For the next question is asking the same question about pathology, but I think I've covered that. How many applicants per place for this course? Would further maths help? I don't actually know how many applicants there are per place. I don't know. I'm just trying to think. But would further maths help? I think if you are embarking on a career in science, maths is certainly important. And, the more maths you do, the better, frankly. I think Gemma would agree with that. Is that fair comment?
Yeah, I would agree. Yeah.
Yeah. But the entry requirements are as stated, and beyond that, it's just extra stuff. If you meet the entry requirements, you will get an offer of a place. Is it useful to start reading research papers in free time before applying? And where would you recommend finding good papers? I think probably it's not useful. I think that you would find them pretty incomprehensible, actually. I think if you want good information, there are a number of sources. Some of the mainstream media have pretty good science coverage. The BBC has good science coverage. The Guardian has good science coverage on its websites. And you'll often find that if a new paper comes out, so I'm reading this paper about flu at the minute, and I actually saw it because I saw a news story that had picked it up. And you can often use those news stories to get to the original paper. They're often open access, so you can read them. I think that you would probably not, as I said, cope with the paper. What you may find, though, is that often these really important papers that are newsworthy, the journal publishes a commentary to go with them. Sometimes it's known as a news and views article. Those are very accessible, and you will usually find those next to the article itself. I'd also say the New Scientist is really good. And if you want one source of information, then that's it. But there's also a lot of sort of independent blog type activities. So I would just encourage you to explore and follow your interests. Because as I said, this whole business is all about curiosity. And the sooner you start unleashing that, the better. What is the ratio between, Leah again, between lecture, lab, other time? And how is it different in years 1 to 3? Right. Gemma, tell me about year 1. I don't really know much about year 1, lab versus lecture time.
Right. Okay, so in year one, it was mainly lecture based, and we had, I think, one lab session a week, and that was
Okay.
2 hours. And then I think we had lectures basically every other day of the week that were like, it was either 1 2 hour lecture or 2 2 hour lectures per day. And then the labs in year one were like quite broad.
Hmm.
I'm trying to remember what we did, but I remember
It's not
one
that long
time
ago, was it?
a lot's happened. Um, I think one time we did like an EKG, and then another time there was like something with cow blood. But yeah, they were quite broad.
It
And
obviously had
then
massive impact. Something with cow blood.
yeah. And then in year two, we also had a lab session, and that was, I think, about the same. It was like 2 hours once a week. And then over reading week, I think we had like 3 4 hour lab sessions, and we did stuff like gel electrophoresis and like cell cultures and stuff like that.
Okay. Okay.
And then year three obviously is your project.
So yeah, so
Yeah.
I can tell you exactly for year 3, which I am involved in, which is that you have between 8 and 12 hours of lecture time per week, 12 hours in the first term, 8 in the second term. That's because you do 3 taught modules in the first term, and each of those taught modules has 2 2-hour slots per week. So you get 12 hours. And the second term, you choose 2 optional modules, same load, so you have only 8 hours. That's by design because you start your project at the beginning of the first term in October, but you're typically learning how to do stuff. By the time you come back after Christmas, you're on top of the methods, you know what you're doing, and you want to have more time to spend on your project. And so you have a lower amount of taught time to open up space to do the project. So the balance is somewhat different between years 1 and 3 in the ways that we've just described. There's also, I should say that the first year, quite a substantial part of the taught content is what we call blended format. So there are short video stories to watch in advance of the in-person sessions. That carries on a little bit in the second year, but not at all in the third year. So most of the content actually is in person, face-to-face type contact in the lecture theatre. Does the course also cover other fields of microorganism technology, bacteriophages for bacteria, CRISPR, and so on for genetic diseases? We don't have a sort of explicit focus on that, but there are lots of students who are using CRISPR-Cas in their projects to engineer, for example, TCR expression in T cells, or to make cells express stuff. And the immunology modules, both the core and the optional modules, when they're considering things like single gene disorders, immune deficiencies, primary immune deficiencies, and how they might be treated, cover things like these technologies, the latest genetic manipulation and gene delivery technologies. So this stuff crops up, but the focus is not so much on the method as the application. I always think that it's the application and the science that's most important, rather than just learning how the thing works. But you would learn that part and parcel with, for example, the module on immunodeficiency. So yes, all that stuff is covered, but of course it's captured more broadly because of our sort of focus on research and the state of the art. If the papers that are coming out and the direction of travel in the discipline is around, I don't know, using CRISPR to repair single gene defects, then inevitably that will be covered in the content. Okay, I think we are almost at the end. We're almost out of time. Leah, the last question goes to Leah. Do you have any other interesting examples of past projects except for COVID and Gemma's? Yes, there are loads of good projects. I should have put a list up. So let me think. We have so this year, students have worked on how HIV spreads from cell to cell, mechanisms of generation of antibiotic resistance in various types of bacteria. They've worked on generating a pan-coronavirus T-cell vaccine. They've worked on the environment within the liver and how that impacts on immune cell function. Someone has worked on a new T-cell phenotype in type 1 diabetes and whether that can be manipulated for therapeutic benefit. Someone else has worked on an autoimmune disease called lupus and has actually discovered a correlation between a certain type of antibody against a particular target and a particular presentation of that disease. So, you know, there are lots of topics and the students are genuinely accessing the state of the art. And in fact, that last project I talked about is being written up for publication as we speak. So that's quite exciting. And I think Gemma's work's probably going to end up in a high impact paper pretty soon. So yes, I'm sorry I haven't memorized all of the projects, but that gives you a sampling of them. All right. Well, thank you all very much for attending this event. I hope you found it helpful. As I said, do consider coming to the actual open day. I might meet some of you there. If you do come to my talk, do come and say hello and tell me that you were at this one. Thanks very much to Gemma as well for joining and giving us insights. And very best of luck to all of you, whatever you do, wherever you end up. I hope you have an amazing experience at university and beyond. That's it. That's the end. Thank you and goodbye.